Well, thanks for having me, and this is a lot of fun for me. It's the first one of these things I've done in a year and a half. I used to do it like every week, and now I've done one of these in person a long time. So, two things. Peter's right. I am not an investment banker either, but some of my best friends truly are investment bankers. Secondly, if Nubar Athia had heard that Peter say that I'm CEO of Flagship Pioneering, he would have had a heart attack. I'm a CEO at Flagship Pioneering, which is different from being a CEO of Flagship Pioneering. And it'll become clear what that means. Flagship's a big ecosystem, about 50 to 80 different companies, depending on how you think about it. And Nubar is the overall, he's the founder and CEO of the whole organization. So I'm a partner in the firm and also a CEO of one of the franchise companies. So what I'm going to do is just try to give you some things to think about more than anything, and I think that will be a lot more fun than having any of us going on for long periods of time, but to actually have some discussion. And Steve Spielberg and I have known each other for an extremely long time. Steve, I hope you take a look at his bio, because he also, like me, has had experience in academia, in big pharma, in government, and in biotech. And we were just talking about at the table, we might be interested in talking about at some point, how different are they? And of course, in some ways they're very different, but what I always say about this is that, two things are the same. Science is the same and people are the same. And so people act like people, no matter where you go, for better or worse. And the rules of science don't change. The laws of thermodynamics, you'll be happy to hear, still apply, even in the government, although many people in government would seem to be trying to deny that at the moment, but it is true. Gravity and thermodynamics and the laws of economics actually apply there too. And so some of what I'm going to tell you is from my 20 years at the NIH, and some of it, what I'm going to tell you is related to my 6 months at Flagship. But even when I was at NIH, I lived in the Valley of Death. Think of it that way. That's the institute that I ran was founded to do that. So very different from the other 26 institutes at the NIH. In case you're not aware, I should actually tell you this. The National Institutes of Health, $43 billion a year, your taxpayer dollars, thank you very much. About 90% of it goes to universities and medical schools across the country. It's the largest funder of basic biomedical research in the world. An amazing record of productivity. It is, however, and I think it should go on being, mainly focused on fundamental research. That's what it does, and that's what it does best. But I'll get into some of the translational issues later. So, okay, so this is the problem, I think, that is the problem of our era. When I was in training as a neurologist 35 years ago, this was not the case. I mean, most of the patients that I saw, I could diagnose with these syndromic, eponymic disorders, whether it's Alzheimer's or Huntington's or ALS or whatever it was, but we didn't understand what caused almost any of these. And it's really hard to treat something if you don't understand what causes it. But over the last 35 years, during my professional lifetime, that has changed utterly. What we know now compared to what we knew 35 years ago is completely unrecognizable. And if those of you spending time in the lab, you think what a lab looked like 35 years ago and what a lab looks like now, and how much a graduate student can produce now versus what they could produce 35 years ago, they're just night and day, unrecognizable. I mean, graduate students are now doing sequencing genomes in a day. That's just totally mind-blowing. And those are stem cells that I could make out of any of your skin, and I could make neurons that mimic what your brain is doing at will. I mean, again, it's just complete, what was science fiction 35 years ago is now, done by high school students. It's just extraordinary. And most of that comes from investment in the NIH. However, However, how are we doing on the health side? Not so good. So we have to be honest with ourselves. If we look at overall health of the U.S. population, it's not so good. And a lot of the trend lines are going the wrong direction. And there are thousands of diseases which have no treatment. Actually, the majority of human diseases is about 8,000 of them, we think. Actually, not sure how many diseases there are. That's an interesting question in itself. But what is known is only about 500 of them have any FDA-approved treatment. So there's about well over 7,000 diseases that have no FDA-approved treatment. And most of these are rare diseases. About 60% affect children. These are the patients that I spend most of my career with, and for the patients and their families, these are absolutely devastating, and they're also devastating on the financial side too. Can you put this in the presentation mode? It won't go into that.
No, I won't.
No. Okay. So here's I love this. It's a little nerdy, so. Forgive me for that, but I just love this because it's kind of a shock and awe slide. So this is the number of diseases that have a molecular basis that is known, a molecular basis. There are a lot of conditions that have more than one molecular basis, but and I always like to say when I graduated from medical school in 1986, which is on the left side of that graph, it was really easy to study for the boards because there were only a couple of dozen diseases that I had to know the cause of, and now there's almost 7,000. I mean, just think about that. Go from a couple of dozen to 7,000 in one professional lifetime. It just never, ever, ever happened before in the history of medicine and science. And if you're asking why this happened, that's the sequencing phase of the Genome Project. That's why that happened. And that's great, except for the 500 with therapy. So, patients, you may know, like diagnoses, but they also like treatments, right? And my own field of neurology is well known for give you a great diagnosis and have a nice day. Geneticists do the same thing. That's my other training. Here's your polymorphism. Have a nice day. And the patients have this annoying habit of saying, "Well, did you forget something? What's the treatment?" And we say, "Oh, we don't have any treatment. You gave me the wrong guy. Sorry." And a lot of medicine is still like that. And it's absolutely heartbreaking. Okay, you all know about this graph? Yeah, Eroom's law. I find very few people outside of this kind of audience have ever heard this. But if you just think about this as monotonically negative productivity growth in an industry over the last 60 years, what would happen if you had any industry, whether you're making drugs or ball bearings or cars, what would happen if your productivity goes down by half every 9 years for 60 years? That's the drug industry. You would merge, you'd go out of business, you'd acquire each other, and the products that you do make would be exorbitantly expensive. And as I just described the pharma industry. Now, this curve has in selected diseases has started to go up, especially in orphan diseases recently. But the thing that I find most striking about this is think what happened in medicine during this time and science, invention of computers, recombinant DNA, MRI, PET scans, just incredible differences. And yet none of them had any effect on productivity. And there's actually been a lot of people who argue that the avalanche of information has actually contributed to the decrease in productivity because there's so many things to think about that people just get confused and you end up on wild goose chases. And I think there's some truth in that. And that's the kind of thing that the current big data, AI, ML craze is trying to address. And I actually like this. I dug up this graph, or this quote from a McKinsey report back in 2001. This was before the completion of the Genome Project, which, in case you missed it, was April 15, 2003. That was the official end. 50th anniversary of this Watson-Crick paper on sequencing, but on the sequencing of DNA. But you notice this, biopharma could go bankrupt trying to innovate based on all of this information. And I think biotech, and I count myself in this, I think we're doing our best to make this come true. In many cases. And it's an interesting thing to think about. Why is this? So when I left Merck, I went actually to NIH, to the Genome Institute, who brought you the Genome Project, and my job was to translate the genome into biological insights and therapeutic potential. That was my job. And it was the greatest job. I often describe it as the greatest job in the history of science, I think, because it was this moment in time where the genome got finished and it got handed to me. I mean, it's just amazing, absolutely amazing. But it was also amazing how overzealous, or well, to use a hackneyed analogy, how much irrational exuberance there was about what would happen as a result of the genome. And I remember thinking this when I'd been at NIH for about 3 weeks. And my boss at the time, and still my boss for 20 years, Francis Collins, he was the head of the Genome Institute at the time. Of course, he's the NIH director now. And he was testifying before Congress, congratulations, Genome Project's done, and all that stuff. And we're watching this on TV, and one of the Congress people asked, okay, Dr. Collins, we're really excited to have you here. It's been 3 weeks since the end of the Genome Project. Can you tell us about all the drugs that have been developed during that time? And I just thought, I'm doomed. I'm doomed. Can I go back to Merck, please? And there's a lot of that magical thinking in Washington, and you see it in all kinds of aspects. But and we could talk about why that is, and Steve and I talked about this. Spielberg and I talked about this a lot. So one of the things that I did, which you may find helpful, is it became clear when I was in this role that most stakeholders, whether they're scientists or physicians or policymakers, congressmen, whatever branch of government they are simply unaware of the complexity of trying to develop a new medical product. They have absolutely no idea of how complicated it is, and that leads to all kinds of mischief when it comes to ideas about drug pricing, for instance, or margin rates, which we could talk about if you're interested. And so it became evident to me and a number of other people who are part of this thing called the National Academy of Medicine Forum on Drug Discovery, Development, and Translation. Some people from NIH, FDA, Jen Woodcock's on it, among other people, Throckmorton, those kinds of folks. A lot of people from pharma, a lot of nonprofit organizations are on this thing. And we decided we really needed to create an accurate portrayal of how this process actually works for educational purposes, but also to re-engineer the system. If you're going to re-engineer a system, you got to know what the math is. If you think about it as a biosynthetic pathway, you have to understand what the pathway is. And so we did that. We crowdsourced it with this group, and you probably can't see some of these names, but John Wagner was he was at Merck, he was at Takeda at the time. Andy Dahlem was at Lilly at the time. Lynn Hudson was at CPATH, probably a lot of you know that. Sharon Terry's head of the Genetic Alliance. Those are these names I'm reading. Russ Altman's at Stanford. And so we published a couple of papers on this. But let me just show you what we did. And it's much better if you have the animation. It's just more shot on here. So just imagine the animation. What we started with is the chevron. And we realized that the chevron itself is a real problem. And the word pipeline is a real problem, right? Because it looks really easy. And if you're a policymaker, what could be simpler? It's just 6 easy steps, right? It's a pipeline. Something goes in at the beginning, comes out the end unchanged, and the progress is only dependent on the diameter of the pipe. That's what a pipeline is. That is about as inaccurate a description of how a drug is made as you could possibly imagine. And yet we all use the term. I don't use the term anymore because I've realized that congressmen, words matter. And people believe that this is a pipeline. And if you say it's really hard, they say, what is the matter with you? You can't get water to run through a pipe? You really have issues. And yet we're trying to tell the story about how much money we spend and how inefficient this is. So we realized we have to change the way we talk about this. And what that chevron actually is and this is a simplified diagram of what it actually takes, what all of us do. And I always say to my what did I do? To my, thank you. To my my academic colleagues, who I love dearly, you're in the upper left corner. And if you think you've made a drug from the upper left corner, you got some things to learn. And this is like a board game from hell. You could be in this for your entire career and these re-entrant loops. And if you look at this, I would encourage you to look at it. I realize you can't see this probably, but they're like this one. There's one right here, one little step. It's called medicinal chemistry. One step. That looks pretty easy, right? I mean, you could be marooned in medicinal chemistry for years. And so, but we have found this useful because it helps in the dialogue. It also helps with, if you're going to plan where you're going to go, you better understand what the map is. And it's extraordinary. I mean, even at Flagship, how few people understand this. Not the big people, not the people at the top, but the people, the RAs, the more junior people, they all come out of academia where none of this is taught. I trained in an incredibly wonderful clinical department and a basic science department. You're never taught where drugs come from, ever. And that's a real problem. So the other thing we did was made this from a paper version into a Google Maps version. So there's actually 10,000 nodes in that Google Map version, and you can access it on the web and zoom in and zoom out, and you can do a sort of localization feature. Where am I? Where should I go next? Particularly useful for people that have done this before. That's really useful. Okay, so this is the organization that, after being at the Genome Institute for 10 years, of translating the genome, there was this new institute at the NIH called Ncats, the National Center for Advancing Translational Sciences, that I became the founding director of. It's one of the 27 institutes and centers that make up NIH. And Ncats' role is really what it says here: more treatments more quickly. The whole idea was to approach the translational problem after the average academic runs out of gas. How do you create a science of therapeutic development so that there are rules? We understand what the rules are. Because if we think about translation, what all this does is it's almost relentlessly empirical. It's a trial and error process, shots on goal. Shots on goal tells you we don't know what we're doing. This is one of our, we have one of these giganto 3 million wells a week screening systems, and I love them, high throughput screening. But at one point I realized if I have to test 3 million different chemicals to find a starting point for any new drug, that is prima facie evidence of the fact that I have no idea what I'm looking for. I mean, if you're looking for a 7-Eleven and you have to go out and drive around into 3 million different turns, you'll eventually run into a 7-Eleven, but wouldn't it be better to have a map? And so what Ncats' job is to collaborate with the public, with the private sector. And that's Steve was actually on our council, among other people. So was Mike Rosenblatt and a number of people in the Stoner, probably a lot of people you know. And we worked on the problems that we all confront every day in making therapeutics, but we don't understand the science. So when I was leaving NIH, I actually wrote a paper that was published in Clinical Translational Science. I'm not going to look at it back in May, and it focuses on what are the rate-limiting steps to making a therapeutic drug or device. And I think, as we all know, they're really the same for the most part, whether you're talking about cancer or Alzheimer's disease or psoriasis. They're pretty much all the same. And these are the things, target qualification, de-risking, predictive efficacy, predictive tox, all of the clinical trial inefficiencies, electronic health records, et cetera. And so a lot of these came out of that mapping process that I showed you, because the thing I didn't tell you is that we also engaged in a traffic map exercise. That is, if you have a map, you want to know which parts are green, which parts are red, and which parts are yellow. And so, the parts that are red are the parts that are most costly in time, money, or risk. And those are the areas that Ncats work. because if you think about this, like again, like a biosynthetic pathway, you want to understand what the rate limiting steps are and develop catalysts to those steps, not the steps that are not. And that's what we were doing. So just an example, which we made a lot of headway on. We, like a lot of other folks, focused on this problem. Why do drugs fail development? You're all aware of the numbers, which are pretty depressing. 80 to 90% of compounds that go into phase 1 are never approved. And why is that? Well, it's mainly for efficacy. Plurality is efficacy, and about a third is toxicity, despite the fact that they were effective and non-toxic in animals. And so the conclusion, shocking conclusion, which I always like to say, was completely robust and reproducible, is that rodents are not humans. We've discovered that it's absolutely reproducible. But we forget that, right? We make 100 million year of evolution diversion into a mouse. And I love mice. I've done a lot of, somebody's not in my backyard. I love mice, and I've done a lot of work on mice. Also on worms and flies and chickens and non-human primates. But in many places, we've forgotten that they're models. They're not the actual thing. And so, well, gosh, maybe it would be possible to make these. So we started this program at Ncats about 10 years ago. And the idea here being, the failure. And the idea was that the models that we'd always used, heterologous expressed GPCR, the tox cell, et cetera, we realized that they're poor predictors of human drug response, and perhaps not surprisingly, but we had no alternative 20 years ago. And we thought, well, gosh, what if we could combine stem cell technology with microfluidic technology, with imaging technology, computing, and make little human multicellular bioreactors that would represent the structure and function of all human tissues, and use those to test for safety and efficacy of new drugs instead of animals. Now, I got to tell you, when we started this, I was a skeptic. I'm a developmental biologist by training, and I just thought this is just nuts. But I'm glad to stand here and tell you I was completely wrong. And it's a whole interesting topic, because I think why I was wrong is really interesting. But suffice it to say that we now have these miniature bioreactors, which are generally the size of a microscope slide or a USB flash drive, for all of these organs, and that's on the Ncats website. And yes, this person's name, the figure, his name is Chip. Couldn't resist. And we actually have built, not we, these are folks at Ncats, but also and in other institutes, but mainly at universities and medical schools all over the country, collaborating to create up to a 12 or 13 organ daisy chain of human tissues. So a little human homunculus, a little microfluidic homunculus. Those are, in my experience, the whole organ. They're kind of a parlor trick, but not all that useful. The ones that are really useful is we have 2 or 3 tissues. If you've got like a small intestine to look at absorption, a liver to look at metabolism, a kidney, for instance, to look at kidney toxicity, and a brain maybe to see whether it gets through the brain-blood barrier. Those things are established. What I just described, that's present today. And it's all been created in the last 10 years. So they look like different things. It's too bad it's not in the presentation mode, because these things all move. One of the things that is really interesting, and again, probably not terribly surprising, is that these systems, in these systems, the cells do not represent what they do in vivo unless their physical, the physical environment is the same. So for instance, up on the upper left corner, that's a lung. And the type 2 pneumocytes in the lung will only differentiate to a fetal state. But in the fetus, the lungs are not expanding and contracting. So they don't differentiate to the mature state. You've got to make that tissue chip expand and contract like a lung would in a living person, and then the cells differentiate and do what they do in vivo. But of course, you can't put that stretch in the bottom of a 10-centimeter plate, which is what we all used to do. And the same thing applies to liver. You don't, when you get flow through a sinusoid, then you get the actual mimicking of function. If you get a heart, if you allow it to beat and stretch, then you get, it's really, really interesting. And teleologically, I don't know why I expect it to be different, because if the heart isn't stretching, the person's dead. So what do the cells have to worry about? Maintaining differentiation state? It's game over. But this is now possible. These things are these are real pictures of real chips. So just as, I don't want to belabor this, but if you just look at, those of your scientists, think about what you used to do with your head in a biosafety cabinet, and realize that things in red, you had virtually none of those things. And so it's really not terribly surprising that the models that we had were not predictive. And of course, we had to go to a mouse or a rat or a rabbit or a dog or what have you. But these are all possible in these kinds of systems. Now, it is certainly true that these systems do not allow you to do everything. Aging, for instance, is very hard. People are just getting, I was just working with NASA actually on, that's another interesting story, but on aging these up to a year. And you can't keep them in culture for that long. The thing that I found most difficult from a drug development standpoint is that, at least at this point, you have to have a hypothesis. You have to know what you're looking for. Find unexpected findings that you can get in a mouse. Sometimes you don't want to get them, right? But the things that you couldn't predict, the idiosyncratic, like you're never going to see in a tissue chip, because you never would have set up a tissue chip to test the drug in the first place. So that kind of, the ability to do the 40 tissues, right, that you do in a typical animal path study are difficult to do with these at the moment, but they can do a lot of other things. Okay, and so we originally developed them with normal tissues, but then it occurred to a number of people, well, gosh, if we're making these out of iPS cells from normal people, what if we develop cells from people with diseases? Could we mimic their diseases? And the answer is yes, you can. And you can reproduce, these are all, you look at these different diseases, some common, some rare, across virtually every organ system. And these have been created as well, and allow you to understand human disease in a way that you just can't if you make a transgenic mouse, for instance, where the mouse has 29,999 mouse genes and one human gene. It's still mainly a mouse, right? But these are human cells. So again, can't do everything, but you can really learn a lot. And this is a great example of this kind of completely disruptive technology of the sort that I think we need. If you're interested, all the data on validation of these things is all in a public database out of Pittsburgh, table number 1. There was actually a great amount of effort to not have these just developed and then published, but not actually be validated. So all of the chip platforms had to be sent to a second place that would see if they actually performed as they were designed to. And all those data go in here. I want to just tell you how this is changing in biopharma and the FDA. The FDA was a partner in this from the very beginning, because of course they have a big dog in this fight, no pun intended. Cedar ended up in there twice in the second salt bill. But anyway, the CBER, CEDAR, and CIFSAN all have internal tissue chip efforts, as well as working with outside labs. The IQ Consortium, that I think probably most of you know about, the International Consortium on Pharmaceutical Safety and Quality. It's a consortium of most of the big pharmas and a lot of medium-sized ones. These are the safety pharmacology, tox folks at the big companies. And there's a MPS affiliate, as they call it, who works on these. They published a whole bunch, these are all pharma folks, have published a whole bunch of publications on the use of these for a whole bunch of safety and indications. And they're working on the other issue, which is disease modeling and all these different modalities. and there's a whole bunch of companies, and this is outdated number, but the number of the big companies have their own internal efforts as well. And there are, again, sorry for not having the animation, but this has been an enormous area of small company birth and growth. I mean, you probably all heard of Emulate, but there are many others focused on sometimes individual organs, sometimes many different organs, and they have both a CRO model or a buy and use model, either or sometimes both. This paper, in case you want to get a quick update on this paper came out yesterday. This was hot off the presses. I made this slide on the train coming down. So it doesn't get any more up to date than that. These two authors are from a Dutch company called Ncats. It's on this list here. But you'll see, again, you might not be able to see this, but it goes through all the steps, fundamental, discovery, target validation, pharmaceutics, preclinical safety, clinical development. Number of places are developing these to do clinical trials on chips to qualify people for clinical trials and to try to exclude people who might have adverse effects. If you really want to understand where everything is, read my paper, which is a rampant self-promotion. This is actually a paper that came out of a Keystone meeting about a year ago, and really does give you the state of the art in everything when it comes to this platform. So that's just one example of the kinds of, I showed you that big, long laundry list of problems, and this kind of technology development is ongoing in every single one of those. And again, if you're interested, that paper I showed you from Clinical Translational Science actually goes through each one of them and tells you what's going on in terms of novel technologies and novel paradigms and novel approaches and who's doing it and what progress has been made and what needs to happen next. So if you want a real fast CME course, there it is. I thought I would just show you one other technology because it's gotten a lot of hype, but I think it is an example of something that is already starting to change how we think. So you're all aware of plain vanilla gene therapy. Take full-length gene, put it into a person. In that case, they still have the genes, the faulty genes that they have. They now have a third copy that substitutes for the loss of function for the other two. And there's been a lot of success. That's Luxturna or the SMA gene therapy. And then there are oligotherapies like Spinraza that also don't change the gene that the person has. Therapies like that act literally like molecular band-aids, and they sit down on top of the faulty gene and cover it up so that the molecular machinery can't tell it's there, and it ignores the mutation. Pretty cool. Again, not something that I think any of us would have predicted, but it works. But the ultimate has always been, well, gosh, can't we just get a spell checker, for heaven's sake? And the body does have a lot of spell checkers. You should know that. That's why we're not all mutants, or all mutants of one sort or another, but no big mutants. And it turns out that it actually is possible to engineer enzymes that exist in nature for a variety of reasons to repair individual misspellings. And it turns out that there may even be this form of this enzyme, that's the crystal structure of it, which 4 versions of which would potentially cover most human mutations, point mutations at least, because you figure this DNA has 4 possibilities, right? A, C, T, G, and it's just a misspelling. The letters are wrong, and so there's a limited number of possibilities of corrections that you want to make. You don't have 26 letters, you only have 4. So a spell checker is pretty easy. So, and it actually turns out that it is possible, it has been shown in mice now, given the caveat that I told you about mice, but still in mice it has been possible, and this was about 6 months ago, it was published, that it was possible to actually correct an inherited mutation in a form of progeria, aging syndrome in a mouse. And so where are we going here potentially with gene-targeted therapeutics? And this is just an example of more mind-blowing work. We've been doing this for the last few minutes here. The first is that we can begin to think of doing for diseases in an intentional way what has happened in every other area of biology, which is multiplexing. They're doing many at a time. High throughput screening, multiplexing PCR reactions. This happens across biology, and yet we still approach one disease at a time, pretty much, as if they're completely separate entities. And of course, they're not. They're all connected with each other. It's all a giant jigsaw puzzle, and they're all connected in one way or another. So you can begin to approach many diseases at a time, thinking about common delivery vehicles, which a lot of people are thinking about, right? So if you think about gene therapy, you got to stand yourself on your head when you're thinking about an excipient versus an API, right? Because the API in gene therapy is the gene you can't patent for the most part. What people compete on is the excipient, the carrier that takes this into the cell. It's really quite bizarre. But conversely, let's say you could come up with a few common delivery vehicles that could deliver thousands of different genes, like what you do with TSA, right? You have a certain small, medium, large will fit in the overhead bin. You got to have the right size. Exactly the same issue. And then you can begin to think about treating hundreds or even thousands of diseases at a time. And this is going on right now. And then, if you take this even farther, you redefine diseases, at least rare diseases, Mendelian diseases now, by their mutation. And so ultimately, you don't care what the gene is. You don't care what the phenotype is. All you care is the misspelling in the word. Think about this as like writing. If there's a T, every place there's supposed to be an E. You just do a find and replace, like you do in a Word document. And believe it or not, so then it doesn't matter what the word is. It doesn't matter what the meaning was of the word and the misspelling. That's the phenotype, the disease phenotype, or the function of the gene. It doesn't matter. You're able to use the same therapeutic potentially for hundreds or even thousands of different misspellings. Holy cow! Now we're cooking with gas, as they say. And the reason this is possible is that there is a relatively small number of ways that DNA can get messed up. And those of you, again, who are scientist types will recognize these splicing mutation, transmissions, transversions, et cetera. It's analogous to what's going on with Keytruda and some other medicines in cancer, right? Where the tissue of origin, whether it's breast cancer or prostate cancer, whatever it is becoming decreasingly relevant. The issue is, what is the molecular defect that leads to cancer in the first place, independent of the organ that it's in. And we're even thinking about, at the IIS, we were thinking about, believe it or not, we can talk about this if you're interested, just-in-time gene therapy. I've actually dreamed about drive-through gene therapy, where we'll probably never get there. But it actually is possible to think about this, where you could diagnose someone as having a mutation, make an oligo, make an ASO to cover that up if you have the right kind of mutation, or a gene editor, give them the gene editor that you know they need, and by the time they go out of the drive-through, they've been treated. So what all those medicinal chemists do, I'm not sure. We'll have to figure out something new. But seriously, we all know that these approaches are going to be most useful for Mendelian diseases. But for the common diseases that I now work on in my new company, I think they're going to be useful as well. So what are the problems? What's going to keep? Well, these regulatory class effects we did a lot of work with the FDA on this. How many diseases would you have to do before you could say that the next one you could come in and use the same CMC manufacturing, chemistry, and the oligo, or the gene therapy, or the carrier, the AV, or whatever it is? How many would you have to do before you wouldn't make the next person do all the CMC and regulatory or the manufacturing and tox and release assays and all that kind of stuff? But FDA is really interested in that discussion. They are an equal partner in these discussions. They are very motivated to make this happen. The problem with gene therapy, of course, what's advantage and disadvantage, is often irreversible, right? So if you put a third gene into somebody, or you edit their DNA, and it turns out to be a bad thing to do, you can't get it back out again. There's no, the PK is infinite, you know. So that's and of course that could be an advantage, but disadvantage too. Off-target effects, of course, can be mutagenic because you can mess around and place with the DNA you don't want to. Getting them to specific tissues and cells is still really hard. It's been hard from the beginning. And manufacturing and CMC on these things is really underdeveloped, particularly for gene therapy vectors. It hasn't changed much since I was in training 30 years ago. And of course, the costs are difficult. You know, $2, $3, $4 million of treatment. Now, over the lifetime, you know, you can amortize that, of course, but the reimbursement models we have aren't well suited to that. You know. Okay, so just in the last just few slides, I just want to give you an idea of what's going on in Flagship. So Flagship, one of the reasons I decided to go to Flagship is it has a lot of the characteristics of what I just described to you of the institute that I ran at the NIH, except they're actually making products to make it, which NIH of course doesn't do. But all the same kind of things that I talked about there. And the thing that is interesting that you might want to look at, because a lot of this is published, is that they've actually tried to make as much of a science of company development as possible, thinking that there have got to be rules for success and failure, just like there have to be for medicinal chemistry optimization or toxicology prediction. And so they have this single business model, so they call it a proprietary process, but it's the barrier to entry is very low because they publish it. But like a lot of things, it's a cultural issue that can be hard, I can imagine, to institute in other places. They're all platforms. All the companies are platforms. There's no single product company, and their culture is really interesting. You notice what they put down here: comfortable operating in uncharted territory, which Nubar, who grew up in Lebanon, he's actually Armenian. His family got out of Turkey during the genocide. He grew up in Lebanon and then came to Canada actually, and then this country. He likes to talk about an immigrant mentality, so-called, of science, that you're used to working in a very unfamiliar environment. And this term paranoid optimism, which sounds like an oxymoron, but I think you all know what I mean. So these are all the companies, and they're either new modalities, things like Moderna or Leron, if you followed them recently, or new biology companies like the one that I run, which is this little tiny right here, we're still in stealth. And again, if you want to read about this, because it really is an interesting model that historically has returned double the average investment. That's why Flagship was able to close a single round that was $3.5 billion back in June. $3.5 billion in one round, and that's because of the historical success. And the historical success comes from this model, this evolutionary model. And so what that's produced now, if you take this sort of funnel, this is what it actually looks like in practice. There's a bunch of these early companies, and then these so-called proto-codes and new codes and growth codes. And Moderna, for instance, is way out here, or Ravello, Denali, you might have heard of them, Sana. And then there's other ones which are earlier, like Generator, Leron, and then there's a bunch over here that they're still in the primordial ooze over there. May never make it out. And just the last thing to tell you is, again, this has all been published, which is a little bit surprising to me, but there's been a whole bunch of HBS studies on this, on Flagship. And this is something that New Baratheon and Gary Pisano, who's a guy at Harvard Business School, published just a few months ago about why they think this model works and why they think its success rate is so much higher than usual. And then just last week, I don't know if this is good or bad, because this could indicate a top. Whenever you end up on the cover of Fortune, that's the end is near, I think, in some ways, but hopefully not. But Flagship got one of these Change the World awards from Fortune. But it does remind me, right before I went to Merck, you remember when Merck was the most admired company in the world for all these years and years and years, and then within 6 or 8 years they cratered. That's an interesting discussion too. But so I think at this point we're going to go to talk to Steve. Yeah.
We've run a little bit over, so what I'd like to do, you can actually probably stay there because.
What is Steve?
How do we have a fireside chat?
If you turn around, you'll see there he is.
Oh, there he is.
If you stand at the podium, you'll be mislabeled as Peter Young, which is most unfortunate.
Yeah, he makes more money than I do.
Yeah, so but we'll have to shorten the fireside chat, which was supposed to be 20 minutes. If we can shorten it to 10, otherwise we'll run.
All right, Steve.
We'll keep it quick.
Okay, great. Okay, great.
Chris, as always, always leaves me with such a sense of excitement about the science. As you can see from my library of analog devices up on the shelves, I graduated from medical school 15 years before you did. So it's I can really appreciate the incredible impact that new science has had. And there's no question but that for those of us in the audience who have been through this and understand the science of it, there's a sense of excitement. But since we're short on time, let me just pose one issue just to get your ideas, and then we need to talk afterwards. One of the things that I perceive, having been a medical school dean, having been a patient, having kids who are physicians, and watching what goes on in the world, is that I've never seen a time in our history where communication in general is so difficult. Much of the language that we share is unshareable, not only with lay people, but for medical school graduates, for new doctors who are out there struggling day to day to take care of patients. Yeah. And one of the things that really is in the back of my mind is the whole issue of how we communicate among those who discover, sponsor, fund, regulate, ultimately use patients and advocates, how we communicate amongst ourselves. And I must admit, I find the COVID vaccine discussions phenomenally disheartening in terms of, and forget the politics, forget even the media that overhype dissension. There is a great deal of lack of understanding. And to me, the lack of ability to communicate leads to lack of confidence. But it also leads to the lack of funding. It leads to the lack of conversion of drugs from reagents into real therapeutics. And to me, miscommunication right now is one of our greatest risks in the risk-benefit equation of healthcare, which to me has very real consequences in terms of lost opportunity for new drug development, but for all of our credibility. The research establishment, patient care, NIH, FDA, CDC, medical schools, hospitals, pharmaceutical companies, large and small. And I worry, just talking to neighbors and friends and those who use healthcare and those who provide healthcare, that this is having a very difficult impact on our ultimate ability to provide individual and public health. So you've been at this game in the same spheres that I have. You presented some of the most delicious science I can imagine that's come out of the last, for me, 45 years of enterprise here. Do you have any thoughts how we translate that into clinicians, patients, advocates? How do we overcome the communication gap?
Mhm. Well, first of all, Steve, I think you're absolutely right, and I think any of us who are frank observers have got to be bothered by this. That at the same time that we have this amazing ability to understand and even treat diseases, we have the very people who we hope to help not trusting us with those therapies when they're developed. And I think there's a complicated question and a complicated answer, as you know, but I think to me, it comes down to a single word, which is staying humble. Really, when you think about the problems that we've run into, it's because people have begun to believe their own propaganda, and they've lost the perspective and the humbleness that if you're realistic, you know Mother Nature reminds you need to have every single day, because Mother Nature, for the most part, does not give up her secrets. And what we think we know, when we know, is far less than what we need to know. And what I've seen, whether it's in politicians or in scientists or in physicians or in public health officials, this sense of absolute certainty, it is understandable and tempting. Of course, through history, it's always been, but it ends badly. It always ends badly, and it's a hard thing to get people in that position to adopt the countenance of. I find that the best way to stay humble is to never stop talking to and working with patients, because patients, TS, not CS, not CE, but I think we need that too. But whenever I think that I've done something wonderful, I talk to a mom of a rare disease patient, and I remember one of them said to me once, I was talking about how wonderful this discovery was, and she said, I love fundamental discovery in my child's disease. I love basic mechanistic discovery, but when my daughter gets sick, I can't give her a publication.
Yeah.
And I thought, oh, that hurts. Because I thought, I'm really proud of everything I've done, and at the time I thought we were, on our way to have a drug. I was completely wrong. And I think from what I've seen, if we partner with these folks, with the people who we're trying to help, then they understand how hard this is. They understand that we're working as hard as we possibly can, and it's expensive and failure prone. But it also, when you let them in on this guild that we like to put ourselves in, this country club, like where we are now at the Yale Club, for God's sake. Steve and I are both from the Orange and Black institution. Surprised I was even let in here. It does wonders, and that's the major thing I see, and I hope will return. And of course, if you see this in, in finance all the time, in this kind of megalomania that always ends badly. Every time I think, I'm getting a little uppity, I go and we rewatch the Theranos special because I think we all say things to our groups that Elizabeth Holmes did, and I was physically ill the first time I watched that because I thought, do I do that? Have I begun to believe my own propaganda? Because at that point you fail, and then you've got a real boy who cries wolf problem, and the public just does not believe you. They just don't believe you because they've been snookered so many times, and I think that's what we're seeing. So, thank you.
It's absolutely right on, and suggests to me a couple of things I want to write with you that need to be written. And, when I look at all the stuff that's gone on over the last years, both the incredible opportunities and the incredible disinformation, this comes back to strike me the lack of general understanding, and it again goes back to humility, that science is not a collection of facts that I have and you do not have.
Yes.
It's a process.
Yes, exactly.
And it's a learning process, and it changes. Every time folks have suggested modification of how we look at vaccines or how we look at other modalities of public health protection, and it changes, to me, that's good.
Yeah.
Because we've honestly looked at the data and it changes.
Yeah.
Those who refuse to look at those changes are those who are doomed. But somehow or another, we have to teach that more broadly. And maybe we can work on it together.
Look forward to it. Thanks, Steve.